All right, so today I'll be taking you through the Mendelian Inheritance Lab. So it's important that before you start this lab, you read through the learning objectives and the introduction. It'll give you a good background as to what we're doing. And then this part right down here, this inheritance problems part, is really important and helpful for figuring out how to start on these inheritance problems and these Punnett squares and Mendelian inheritance problems that we'll be doing in this lab. Make sure to complete the pre-lab questions. Those are all based on the background, so after reading the background you should be able to answer the pre-lab questions, and you can even pause this video if you need a second to do that. And then when you get to this page, this is where we'll begin, and we'll start the answer sheet. And so just remember on this lab that a lot of these problems are hypothetical problems, and when we're talking about genetics in real life, a lot of times it's a little bit more complex than this, but these problems give us an idea of just how Mendelian inheritance can work. All right, so let's begin here. We'll start on the first one. It says in humans, 6 fingers is the dominant trait, 5 fingers is the recessive trait. Assume both parents are heterozygous for 6 fingers. So the first part of this question asks, what is the phenotype of the father? So phenotype here is the physical characteristics. It's not always a visible characteristic, so it could be blood type, for example. That would be a phenotype. It is the characteristics of, in this case, the individual, and so the phenotype would be either 5 fingers or 6 fingers. And in the problem, it says that both parents are heterozygous for 6 fingers, and so for this one, we would write that the father has 6 fingers. So next it says, what is the phenotype of the mother? And again, for the same reason, phenotypes of physical characteristics, the phenotype for the mother is also going to be 6 fingers. The next part, part C, says what are the genotypes of the parents? So for this one, genotypes are the genetic makeup of the parents. And when we're talking about genetic makeup, we are looking at the combination of alleles. So alleles are the gene copies that you get from your each parent, so one from mom, one from dad. A lot of times we write these as single letters, either a big letter or a little letter. So the combination of alleles could be one big letter and one little letter. In that case, it would be a heterozygous individual. Combination of alleles could be 2 big letters. That would be a homozygous dominant. Or the combination of alleles could be a little letter, little letter. In that case, that would be a homozygous recessive. And so the question again asks, what are the genotypes of the parents? And back to the problem up here at the top, it tells us that the parents are heterozygous. So each parent is going to have one big F and one little f for their genotype. The rest of the question says, set up the cross and use a Punnett square to determine the genotypes and the phenotypes of the children. And so we're going to go ahead and do that over here. We'll put one parent on the top of the Punnett square, and we'll put one parent on the side of the Punnett square. And then we're just going to bring those alleles down, and these are our possible combinations in the offspring. So you could have big F, big F, little f here. or little f here. And so then part D, right down here, it says, what is the probability of a 6-fingered child? In this case, 6 fingers, we said, was going to be dominant, and so that would be a 6-fingered child, 6-fingered child, 6-fingered child. So we would have 3 out of 4 possibilities, or 75% chance of having a 6-fingered child. Part E says, what's the probability of having a 5-fingered child? And in this case, our 5-fingered child would be little f, and that would be 1 out of 4 possibilities, which would be 25%. If you'd like to pause the video at this point, you can try question number 2. Question number 2 goes right along the same lines as the first question that we just did. It says, if the father is heterozygous for 6 fingers, so we can set up the father here. He's heterozygous, so he has 1 big F, 1 little f. And then it says the mother has 5 fingers, meaning she would be little f, homozygous recessive. Then we bring down each of the possible alleles. Big F, little f. And we find that we have 50% and 50%. So 2 out of 4 would equal little f, which is 5 fingers, which equals 50%, and 2 out of 4 are big F, little f, which equals 6 fingers, and that's a 50% probability. And I should say here too that, remember, this is a 50% probability for each child that this couple would have. That does not mean that they're going to have 4 children, and if they have 4 children, there will definitely be 2 children born with 5 fingers and 2 children born with 6 fingers. It's just a probability. So that's the probability of each child that this couple would have on this pairing. For question number 3 introduces a new inheritance pattern, and that inheritance pattern is incomplete dominance. In incomplete dominance, you get a third intermediate phenotype. Remember, physical characteristic, phenotype. And in this case, it is in the heterozygous genotype. And so our incomplete dominant trait would be, in this case, where we have a heterozygous pink flower. So we have a homozygous red flower, that's big R. We have a homozygous white flower, which is big W. And then our heterozygous, our intermediate third phenotype, is our pink flower. And so the way that we set up this Punnett square is we want to go ahead and, it says a cross has been made between 2 pink flowers. And so we can set up our Punnett square in the same way. We'll draw our boxes. We're going to put 1 pink flower on the top here, and we're going to put 1 pink flower here on the side, and then we're going to bring those alleles down into our possible combinations. And then we are going to get 2 out of 4, which would equal our 50% WR here. Actually, I'm going to write this on the other side where I have a little bit more room. So 2 out of 4 equals 50% WR, which is our pink color. We are going to have 1 of 4 equal to 25% our big W, which is our white flower. And then we're going to have 1 of 4 left, which equals 25% big R, and that equals our red flower. So again, this is an example of incomplete dominance. And in question number 4, we're going to use incomplete dominance again. And this is going to be similar. So if you want to stop and pause the video right here and work on this on your own, you can. And I'll go ahead and solve this one. So in this case, we want to set up our Punnett square again. It says a cross is made between a red flower. We'll write the red flower down. Big R, and a pink flower. Pink flower is going to be big W, big R, and then we will bring those alleles down for our possible offspring combinations. And we get 2 of 4, or 50% WR, which is our pink color. And we get 2 of 4, or 50% big R, which is our red color. All right, for the next problem, these are double factor or dihybrid is what they're called, inheritance problems. And so in this case, we are looking at 2 independent traits that combine independently through Mendel's law of independent assortment, and then recombine in our offspring to give us some different traits. And so the way that we solve this is given kind of step by step in question number 5, and so we'll go through that step by step for question number 5. So the first part says assign a symbol for each allele. And so in the problem it says in humans the allele for free earlobes dominates the allele for attached earlobes. So let's take that apart, and I'm just going to make, you could pick whatever letter you would like. I'm going to pick a letter E to stand for earlobes, and so it'll either be a big E or a little e, and that's going to be our earlobe allele. And we want to pick a allele for our hair color. So I'm just going to pick H for hair, so a big H or a little h, and that's going to equal hair color. Okay, and again, these are hypothetical traits and alleles, and so hair color is actually much more complicated than just a single gene, but it's a good example that we can use for a dihybrid cross. So that means in earlobes, big E, or big E, little e, is going to be free earlobes. And then little e is going to give us attached earlobes. And that means for hair, a big H, homozygous dominant, or big H, little h, is going to give us dark hair. And a little h is going to give us light colored hair. All right, the next part says determine the genotype of each parent and indicate a mating. Remember, genotype is the allele combination. And it tells us in the problem up here that our parents are heterozygous. And so we are going to write down one parent that's heterozygous for earlobe, and they're also heterozygous for hair color. And then the other parent is heterozygous for earlobe, and they're also heterozygous for hair color. And we're going to just go ahead and write those right next to each other, kind of as 4 letters. It says to indicate a mating, and the way that we normally do this is just by putting a cross between the two, or an X between the two, which just means a cross. And so the next part says determine all the possible gametes for each parent can produce. Remember, a gamete is a sex cell, so if this is humans that we're talking about, which we are in this problem, it's going to be sperm or egg cells. And in these, in gametes, remember, gametes have half the number of chromosomes. And so they're only going to have one allele or one gene copy per sex cell. And this is really important when setting up a dihybrid cross, that you remember this. And so in this case, we would just have one letter E, that's one allele, and we can have one hair allele. So I'll just pick a big H right here. Or the parent can have a little letter E for earlobes and the big letter H for hair. Or the gamete that could be produced by the parent could be a big letter E and a small h. Or we could have a little letter E and a little letter H. But those are the only 4 possibilities that each parent could produce. Now for part D, you're going to run into a problem because you're not going to have enough room to work out this Punnett square. So you might want to grab a separate sheet of paper or a scratch sheet of paper. I'm going to move over to the side here. It says determine all of the possible allele combinations that can result from the combining gametes, and we're going to draw and use a Punnett square. Now this is going to be a little bit different Punnett square. In this Punnett square, this one is going to be twice the size of our normal Punnett square because we have 4 gametes in this case. We have 4 sex cells that we're crossing, and so we've got to draw a 4x4 box rather than just a 2x2 box. And so then we're going to take our parent allele combinations and we're going to put them right over here. So big E, big H, little e, big H, big E, little h, and little e, little h. And it does say in this cross that both parents have the same genotype, so we're going to put the same genotype on the side here. Little e, big H, big E, little h, and little e, little h. And now we're going to go ahead and fill this thing in. It's going to take a little bit of time. And you know what? I'm actually going to let you take a minute. If you want to pause the video, you can finish filling that in. And what you should get when we figure out the rest of the Punnett square is you should get a number of genotypes. Remember, our genotypes are our allele combinations. And then our phenotypes are going to be the physical characteristics that these correspond to. So either attached earlobes, free earlobes, or dark hair or light hair. And so in this box, we can write down each of those possible combinations. So our most common combination after we finish this is going to be our free earlobes with our dark hair. Those are both dominant traits. We're going to get some with attached earlobes and dark hair. And we're going to get some with free earlobes. We'll zoom in a little bit. And light hair. And we're actually just going to get one with attached earlobes and light hair. So once you fill all this in, you should be able to then transfer the genotypes here. See, this is big E, big H, or sorry, big E, big E, big H, big H. Those are both dominant. That's one of our free and dark possibilities into the genotypes. Okay, go ahead and transfer all of the rest of these into these boxes. When you do, you should notice you're going to end up with 9 different genotype possibilities in this box. You're going to end up with 3 in this box. You're going to end up with 3 in this box. And you're going to end up with just 1 combination, which is this little e, little h combination. Let's look and see where that would be. That would be right here on our Punnett square. You're going to transfer it. That's the only one with that genetic combination right there. You're going to transfer that right over. For question number 6 is also a dihybrid cross. So it is going to be a lot like question number 5. And I'm going to go ahead and allow you to do that on your own. And then you can show that completed when you turn in your lab. But I'm going to let you do that one on your own. And the question at the end is, can this couple have a child with normal vision and no headaches? And if it's yes, show me where that possibility is in the Punnett square. And I'll give you a hint. The answer is yes. You'll just need to show me where that is a possibility in the Punnett square. For the next problem is a sex-linked problem. And the inheritance patterns for sex-linked problems are a little bit different. And so you probably already know, we'll do a little bit of background on this question, but you probably already know that a human male is going to have a X chromosome and a Y chromosome, and a human female is going to have a X chromosome and another X chromosome. In this case, when we're talking about color blindness is a trait that is found on the X chromosome. And so if a male has, sorry, switch to red here, but if a male has a color blindness gene, we're just going to write that as XC for color blindness. That male's genotype is going to be at this XCY. So that's going to be a colorblind male. A colorblind female, on the other hand, is going to have 2 of these mutations. And so this is the reason why color blindness runs it's about 1 in 20 in males, and it's about 1 in 200 in females. It's much more common in males than it is in females because they have to inherit both mutations from both parents. So colorblind female would be XCXC. Females could also just have 1 X, normal X, and 1 colorblind mutation on the other X. And in this case, we would call her a carrier for color blindness. So she would be a carrier female. So in our problem now, it says that a normal male, so we'll draw our Punnett square, go ahead and put our male here on the side. normal X, normal Y, marries a colorblind female. So we'll put X's there with the C. Now we're going to go ahead and complete our Punnett square. And if we look at our initial Punnett square, we find that 2 of 4 equal XC X, which would be 50% carrier female, and 2 of 4, or 50%, would be colorblind male. But we want to go back to the question and look at what the question's asking. It's not just asking for the percentages. It says if they have a daughter, what is the chance she will have normal vision? So when we look at percentages then, if we want to look at the daughter percentage, we just have to look at the females. So what is the chance that she will have normal vision? Well, that is going to be 100%, because if you have a female, that daughter will have normal vision, even though she will be a carrier. She'll be a carrier if female. The next part of the question says, what about a son? So if male, we're only looking at these percentages down here. And so if male, then we are going to have a 100% chance of having color blindness. Question number 8 introduces another inheritance pattern. It's called codominance. In codominance, you have multiple alleles or gene copies that combine to give new traits. So a great example is blood type, which is our example for this problem. In blood type, I'm just going to make a little chart of genotype and phenotype here. Where the genotype, remember, is the genetic combination of alleles. And so phenotype would be the 4 different blood types, just ABO blood types, so type A. And for the genotype, you can get A-type blood if you inherit 2 A alleles or an A allele and an O allele. For B-type blood, you can inherit 2 B alleles or a B allele and an O allele. For a B-type blood, you would inherit both the A allele and the B allele. And for the last one, I'm running out of room. I'll put it over here. Last one would be, the phenotype would be O-type blood. And the genotype for that one would be OO. You would inherit 2 O's. And so for our Punnett square, we're going to set up for this problem. It says a man with alleles for blood types A and O marries a woman with alleles for blood types B and O. And so we're going to complete our Punnett square. And then it says list the types of their blood, list the types of blood their offspring could have and the probability of each blood type. So we'll start with AB. AB here is going to give us AB type blood. And since there's 1 of 4, that's going to equal 25%. There is a BO combination, which is going to give us B-type blood. And there is 1 out of 4 for that one. So we're looking at another 25%. there is AO combination, which is going to give us an A type, which is 1/4. It's going to equal another 25%. And then we've got our last one, our O-type blood, which is another 1/4 and equal to 25%. So the last problem here requires a little bit of detective work, and so we're going to figure this out together. It also introduces a new inheritance pattern, which is epistatic. Epistasis is when, in this case, one gene stands over or stands on top of another gene's expression. So in this case, our example is albinism, and so an albino is a person with no pigmentation in hair or skin color, and if they have the homozygous recessive genotype for albinism, they're going to have the albino trait or phenotype, and that is going to stand over their normal hair color. So you're not going to be able to tell what the original hair color was, because the new hair color is going to be white. And so in this problem, it tells us first, and we want to go ahead and define the genotype and phenotype here. It tells us that a homozygous or a heterozygous and a homozygous dominant genotype is going to give us normal pigmentation. It tells us that a recessive homozygous phenotype is going to give us albino pigmentation. And then it tells us that big D, or big D, little d heterozygous is going to give us dark hair, and little d is going to give us light colored hair. And so in the problem, it says two parents, both with normal pigmentation, one with dark hair and the other one with light hair, produce a child with dark hair and normal pigmentation, a child with light hair and normal pigmentation, and an albino child. So we're going to go ahead and fill in as much information as we know at this point into the chart. And so we're going to start here with the, since we know the light hair parents' genotype, we're just going to fill that in on this side of the chart. So light-haired parent is going to have a light hair color, and that light-haired parent is going to have a little d genotype. We know that the dark-haired parent is going to have dark hair color and is has to have a either a big D, or a little d genotype, because they're both dominant. Second, it tells us that child A has dark hair and normal pigmentation. That's already filled out for us in the chart. And so we'll go on to child B. Child B says they have light hair and normal pigmentation. And so for light hair, would be our little d genotype. For our normal pigmentation, we're not quite sure yet. We're going to leave that blank because we're not quite sure if that's going to be a big A or a big A little a. Oh, we forgot to write in our light-haired parents because we can tell their hair color. They're going to have normal pigmentation. What we're trying to figure out is what their genotype is going to be. And so we'll write some question marks right there. And then for child C tells us that child C is albino. So we're going to write albino here. And we know that the genotype for albinism is little a. So we're going to write it in right there. And then we're still trying to figure out, we're not going to know what hair color the child would have inherited because the albinism gene is epistatic. But we can tell some information now about our parents based on our kids. So because we know that one child was albino and one child was normal, we know that both parents have to have at least one recessive allele. One recessive allele that one parent gave to the albino child, and the other parent gave the other recessive allele to the albino child. If they have normal pigmentation, the parents, then we know that they had to have a dominant allele. So we have just figured out our parents' genotype. We also know that child B had to have had at least one dominant allele. So it must be big A, little a, or could have inherited both big A's, each one from each parent. And since our question is only asking about the genotypes for the parents, I think we have adequately figured out the genotypes for the parents. Oh, one more thing. The dark-haired parent, oops, this was a mistake. That shouldn't be little d, little d. The dark-haired parent must have had to be heterozygous because they had a light-haired child. So the light-haired parent would have donated one recessive allele, little d, and the other one would have had to donate that other one. And so we know that the genotype of the light and dark-haired parents are given right there too. All right, so that's going to conclude the lab. Hopefully this was helpful, and I'll see you